So today we'll be starting with the discussion that of the contents that you guys have studied in the week 3 of the course Experimental Biotechnology. So I'm Rishabh and let's start. So the first question is the most appropriate technique to study the DNA protein interactions is Western blotting, SGS page, gel mobility shift assay, or hydrophobic interaction chromatography. What can be the correct answer? So here the correct answer is gel mobility shift assay and how it's done. So what we do is we prepare a mixture of DNA that we have to study whether it's binding to a protein or not. For example, there is a transcription factor that generally bind to DNA for the uh, production of the RNA in basically transcription. So and this is the DNA molecule. So first what we do, we incubate this particular protein or the extract that we have isolated from particular cell line or an organ. We incubate this particular protein with the DNA and then we ran on a page gel for checking whether the DNA has been bound to the protein or not. So as we can see here, in the first case, we do not run anything. Okay, that is the the supercoiled DNA or very small fragment of DNA that will run as a running control. And then in the second lane, we can see that only DNA has run and it forms a band here. Okay, and in the third case, we run the complex of DNA and protein that has been incubated before, and if the protein would have bound to the DNA, it will run slowly as compared to the DNA alone that you can see here. Okay, so because if we see that DNA has a molecular weight of mg, okay, and DNA plus protein will have a high molecular weight, let's say MPD. So MPD will be greater than MD. That's why it will run slowly as compared to a DNA molecule only. Okay, so it will form a band that is above the particular DNA molecule. Hence, it can be seen that this particular protein has bound to the DNA. Okay, as can be seen from the image itself. So this is a protein molecule. Okay, two proteins have been bound, and this is also a protein molecule that have been bound, and this is a bare DNA. Okay, so this is how we see a shift in the binding of the proteins after the DNA has been the protein has been bound to the DNA. Okay, so this is how a gel mobility shift assay has is done, and this is how it is analyzed. Now, talking about the other techniques, how we cannot use the other techniques in DNA protein interaction. So, Western blotting. For Western blotting, first we have to run a SGS page. So SGS is itself a denaturing or substance, is a detergent. So it basically denatures or removes all the interactions between the DNA or and the protein. So this cannot be used at all. Then we have the SGS page. So this is also just like I said. And then hydrophobic interaction chromatography cannot be used in this case because of the charged molecules. Okay, So here we are using charged molecules only like DNA. So it cannot be used. And if we are using any hydrophobic proteins, then this particular chromatography can be used in the uh, detection of the complex bound with the protein. Okay. So do you have any doubt in this particular question? So if not, then we'll move on to the next question. So here we are talking about the gel mobility super shift assay that can be done by the options are incubating with five folds more protein that required for binding, introducing specific antibodies to binding buffer, increasing the runtime of the gel, or performing the electrophoresis in high voltage. So what can be the correct answer? Yes, the correct answer is 2. Okay, so we can use the super shift assay by introducing specific antibodies for the particular proteins. So here first what will be the reaction? First we have to incubate the DNA with the protein, and then what we'll do, we'll incubate this particular antibody with the protein so that it will have a super shift like we have seen earlier. Okay, so if we can see here, if the DNA molecule is forming a band here, and this is the band in which DNA plus protein is there, and if we add antibody to this particular form, this will form a band here. This will be okay. So this is called as a supershift assay. Why it is done? Because if there is a mixture of proteins, okay, so we cannot detect whether that particular protein only is binding to the DNA. So to increase the specificity of the reaction, we'll introduce an antibody of that particular protein, which is of our interest. So now we'll incubate it with this particular complex and run on a gel. So this will cause a super shift that will allow us to detect, okay, this is the particular complex that we are looking for. Okay, so this is the major function of the super shift assay. So any doubts in this question? Okay, then we'll move on to the next question. So the atom radioactively labeled in DNA to enable us to visualize it in an autoradiogram is magnesium, sodium, lithium, and phosphorus. What can be the element? So yes, it's phosphorus. How? Because if we consider as a DNA, okay, so it has mainly three components. First is the uh, base, that is the nitrogenous base. Okay, then we have the sugar, and last is the phosphate molecule. So it is formed in such a way that this is the sugar molecule, followed it is connected to the base, and here three phosphate molecules are joined. So this is alpha, beta, and gamma. Generally, all the three or the gamma phosphate is yeah. So the gamma phosphate is being labeled by using P32 isotope of the phosphorus group. Okay, so that is why in DNA particularly we use P32 as a radioactively labeled group to label the label the DNA molecules of our interest or to study the DNA. Okay, so this is the phosphorus is the correct answer here. Now moving on to the next question. The blocking agent used to prevent non-specific binding of protein during footprinting assay is bovine serum albumin, salmon sperm DNA, DTT or PMSF. Okay, actually the correct answer is salmon sperm DNA. Okay, DSA is used generally in either immunohistochemistry techniques or sometimes in Western blot also. Okay, salmon sperm DNA has high specificity. Okay, that is why it is used as a blocking agent in footprinting assay. So here the correct answer is salmon sperm DNA. DTT is it is called as diethiotritiol, and it is also a type of reducing agent like beta mercaptophenol. And PMSF is a kind of buffer that is used generally. It is kind of a growth factor that is used in buffers for this particular experiments. Okay. Now moving on to the next question. What is the role of T4 PNK in gel mobility shift assay? So PNK is polynucleotide kinase. The options are it reduces the random binding of the radioactive label, it prevents metallic ion contamination of the gel, it okay. So actually it is a standard protocol. So salmon sperm DNA is used for blocking only in case of footprint assay. In other cases, like for Western blot, the BSA can be replaced by FBS. Okay, so in that case, different yeah, in that case, different molecules can be used as a blocking agent. But in this case, salmon sperm DNA has been optimized to use as a blocking agent. Am I clear? So the role of T4 PNK, that is polynucleotide kinase, in gel mobility shift assay. It reduces the random binding of the radioactive label. It prevents metallic ion contamination of the gel. It catalyzes the sense and antisense DNA binding and catalyzes the binding of radioactive ATP to DNA oligomer. What can be the correct answer? Yes. So what happens? PNK is a polynucleotide kinase. Okay. So it catalyzes the basically addition of a nucleotide at the 5 prime base. Okay, so what we do is generally in case of our reaction and the buffer that we use, we keep the DNA, it has a 5 prime phosphate group free. So now the labeled nucleotide is there, that can be ATP or anything that has been labeled with P32 radioactive element, and then it basically replaces or adds this particular nucleotide at the 5 prime end. So that's why whenever we see in the books also, this is the labeled part, and this is done by the polynucleotide kinase, and this can add the nucleotide respective of the complementary sequence. Okay, and generally it is isolated from the T4 bacteriophage, and that is being used in the recombinant DNA technology from very often. Okay, so the correct answer is option number 4. Okay, so any doubts in this question? So let's move on to the next question. In supershift assays, that is added to bind to the protein prior to forming DNA protein complex. Yes, this is a repeated question. So the specific antibody. So generally we add 2 micrograms of the specific antibody to the particular DNA protein complex to deduce whether the same protein has been bound to the DNA that is of our interest. So the correct answer is option number 4. The cellular extract you will use to identify the transcription factors for a given promoter is membrane protein extract, mitochondrial extract, nuclear extract, or cytosolic extract. What can be the correct answer? Okay, protein extract. So, where are the transcription factors present inside the cell? So, this is the cell, this is the nucleus. Okay, here we have our DNA and all the replication, transcription, recombination, these all processes takes place in the nucleus itself. Okay, so if we talk about transcription, all the transcription factors will be present inside the nucleus only. So if we have to study the transcription factors binding to the particular promoter, okay, so then what we'll use the nuclear extract. Okay, so this is the case. We'll use nuclear extract in this case. Okay, cytosol would not have any of the transcription factors. Okay, rather other proteins would be there like chaperones and other transport and proteins or enzymes would be there. Okay, in case of mitochondria, we would have some of the transcription factors that would bind to the DNA present in the mitochondria itself. So the question would be specific in that case, but here the nuclear extract would be the correct answer. Okay, so any doubts in this question? Okay, so let's move on to the next question. The most appropriate column to use to purify the labeled oligonucleotides for a gel mobility shift assay is Cephalose AEC, Cefadex A50, Danix Carbopac PA1, or Cefadex G50. Can you guess? Okay. So, in case of if we have to separate any labeled or any of the DNA molecules, okay, so then in that case we use Cepadex G50 for their separation. Okay, So this basically is based upon the gel filtration chromatography. So that is based upon the size. And this particular case of Cephadx, sorry, so this separates the DNA molecules that have a size of less than 30,000 Daltons and more than 1500 Daltons, to basically separate them from the small DNA molecules. So that is why Cephadx G50 is used. Okay. So this basically depends upon the gel filtration chromatography. This Q Cepharose is basically a quadruple amine that depends in the ion exchange chromatography. DEAE, this is diethyl aminoethyl Cephadx A50. So this is used in hydrophobic chromatography. But here we want to use it for size exclusion chromatography based upon the size. So that's why we use Cephadx G50 for separation of the labeled oligonucleotides. So are there any doubts? Okay. So let's move on to the next question. Addition of antibodies into the gel mobility shift assay would result in breaking of DNA protein complex, retardation of antibody band, no effect on DNA shifting, or super shift of the DNA band. So what can be the correct answer? So the question is being asked in a different form. So what happens uh, whenever we add an antibody, just like I said, so it will form a band above of the so this will form a band above of the DNA protein band that has been formed. Okay, so this is called as a super shift because how it is named. to- For example, this is the DNA band alone. This is the shift after the protein binds, and this is a super shift after a double protein binds. Okay, so that is why it is called as a super shift of the DNA band rather than a retardation of the antibody band, because retardation means that it will stop. Okay, it will not move at all. So that's the main meaning of the retardation that has been explained. Okay, so this is called as a super shift of the DNA. Okay, so as you can see, as we have seen in the previous case also, like the figure, this band is for the DNA band, right? If we add a protein that binds to this particular DNA, this will be the protein plus DNA band. That is basically shifted from this region to this region. Okay, now if we add another protein, or suppose an antibody, antibody is also a protein, right? So this particular band will shift to upwards with protein plus DNA plus antibody. Okay, so this is called as a super shift after we add a particular another protein that binds to this particular protein, and this will show a band above of this band. That is why it is called as a super shift. Okay, retardation means that it will stop, it will not move at all here. So that's why retardation is not the correct word to use here. Here the super shift would be the correct word in with respect to the DNA that we are talking about. Okay, that is why here it is called as super shift of DNA. So am I clear? Okay, so let's move on to the next question. So this is a analysis based question. Okay, so a researcher was working with three proteins A, B, and C. Okay, which may have potential roles in gene expression. So they basically bind to the protein. In order to validate the hypothesis, EMSA, that is electrophoretic mobility shift assay, was performed. The purified proteins were allowed to bind with the labeled DNA, and the results obtained after autoradiography are shown below. The following observations are made. Protein A possesses the DNA binding motif. Okay, so these were the observations that have been observed. So protein A has a DNA binding motif. Protein B uh, possesses a DNA binding motif. So from the graph, we have to see how what are the options that are correct. Okay, so just think about it and then we'll discuss. So here the plus means that that particular protein is present in that particular lane, and minus means that that particular protein. For example, here all the three proteins are absent in this particular lane, and here only B is present because the plus is there. Okay, that's how you will reduce this question. Okay, let's other also solve. So how we solve these particular questions by looking at the image that they have shown. In first case, all the three proteins are absent. Okay, so this is the DNA band. So DNA moves from negative to positive charge. So here is the positive charge. This is the DNA band over here. Now, if we add the B protein, no band is observed in between, and only the DNA band is observed. That means B protein do not bind to the DNA binding motif, or B do not have a DNA binding motif. Okay, so here the second option is false directly. Now, the same case when C protein is there and DNA is there, so in that case also the band is not observed here. Okay, so that means C also do not have a motif that binds to the DNA. Okay, So in that case, the DNA is only present. So C also do not have a DNA binding motif. Now, when we add only A protein, then we see a band that is shift in the band from the original DNA band. Okay, so in that case, we can say that a particular protein binds to the DNA and has a DNA binding motif. Okay, so here the first option is correct. Then if we say that okay, in second case we add both A and B proteins. Okay, so A also is there, B also is there, but C is not there. So in that case we see a super shift. Okay, so what the super shift determines that this is the DNA molecule, this is A protein, and to this complex or to the A protein, B protein is being bound. Okay, so this causes the super shift. So here the B protein has a site for binding to the A protein. Okay, so the third option, that is protein B binds to the DNA protein A complex, is correct. Now, when we add the B and C protein without the A protein, then we are not able to see the band here also. Okay, so that means both the proteins might bind to the complex only when the A is bound to the DNA molecule. Now, the third case is to check whether C protein binds to the A protein or not. Okay, so in that case, what we do, A protein A protein will bind to the DNA, and then we add C also. In that case, super shift is not observed at all. So in that case, we say that C do not bind to the protein A. Okay, and it doesn't have a DNA binding motif also. So this option is also wrong. Option 4. So the correct options are 1 and 3. So the correct answer is option number 2. Okay. So any doubts in this particular question? Okay. Then let's move on to the next question. So during gel mobility shift assay, DNA protein complex is resolved on 0.8% agarose gel, 5% non-denaturing polyacrylamide gel, horizontal page, or 5% denaturing polyacrylamide gel. What can be the correct answer? So, whenever we talk about any of the interactions that are non-covalent in nature, okay, so in that case, we do not use, here we are talking about the DNA protein interaction. So, that can be van der Waals or anic interactions. Okay, so whenever we talk about any interactions, we will not add the denaturing agents here. Okay, are not added because these will denature all the interactions that are present there between the DNA and the protein, so that we'll not be able to see the interaction between them. Okay, so here we'll be using non-denaturing polyacrylamide gel, that is 5% in nature, to allow the proper movement of the complex. As I have said earlier also, because protein when binds to the DNA, it will increase the molecular weight, so it will retard on the gel. Okay, so that's why it will be 5% non-denaturing gel. Denaturing will not use. Agarose also will not use because of its strength, or the because it doesn't have a strength as compared to a page gel. Okay, and it cannot be run on a particular direction. Okay, so is this question clear? Okay, so let's move on to the next question. Which of the following techniques is used to find genomic binding sites of a known protein. 2D electrophoresis, DNA footprinting, native horizontal page are gel mobility shift assay. What can be the correct answer? So here the correct answer would be DNA's footprinting. And what do we do basically in footprinting is we generally isolate a particular genomic DNA, and then we incubate it with the particular protein. With the particular protein, and then we add the particular enzyme that is DNase. Okay, so what will happen? For example, this is a DNA molecule, and here the protein or the, for example, DNA polymerase or RNA polymerase has been bound to this particular region. So if this is bound to the DNA, this region will be protected by the polymerase or any protein that is bound. And if we apply now DNase, now it will start chopping the DNA from here. If the DNA is bind to this particular protein, it will help mask the DNA from the effect of the DNase. So now this region will not be cleaved by the DNase molecule, and then from here the DNase molecule will start cleaving. So now, what will happen if we run these things on a gel? Then, in that case, if the DNA is not bound to the protein, it will form a ladder kind of structure. And if the DNA has been bound to that particular protein or the DNA, then the region that has been bound will be left like this. And then, what we can do, we can just extract this particular DNA, sequence this, and then we'll know the sequence by to which this particular protein is being bound. So this is called as a footprint of that particular protein on the DNA. Okay. So now let's move on to the next question. The enzyme used in footprinting assay is DNA polymerase, DNase, RNase, or DNA ligase. Yes. So the enzyme that is used in the footprinting assay is the DNase molecule, because that is only the molecule that helps in chopping of the DNA. Okay. Now, moving on to the next question. Which of the following indicates steps involved in DNA footprinting to monitor interaction of DNA with a protein? So, these are the options. Please select the correct option. Okay, so let's see the options first. DNA plus protein, then we'll treat it with restriction enzymes, then run on an agarose gel and stain with ETBR and viewed under UV light. No, this will not be the case here, because the restriction enzyme cuts at a particular site. Okay, so if the protein has that site, then the restriction enzyme will not cut. But if the restriction site would be free, then the restriction enzyme would cut, but that would not reduce the problem, because if suppose this is the restriction enzyme site and the protein binds here, then also it will make a cut. So this will not solve our problem of reducing the sequence to which the protein is binding. So the first option is wrong. Second, label one of the DNA strands with the radio label. Treat one portion of the labeled DNA with the protein of interest, followed by the reaction with DNAs. Then treat second portion of DNA with the only DNAs. Run both the treated samples on a sequencing gel. So yes, this is the correct answer, because as I have said just now, okay. So this is a particular gel that is seen. Sorry for the contrast of the particular image, but as can be seen here, these this would be the strand that has been cut fully by the DNAs, because here the ladder kind of structure is being formed. But in the same other scenario, when the increasing concentration of the protein is being used, so here you can see some of the ladder bands here, or the DNAs bound DNA, and then from here you cannot see any band. That means that protein has been fully covering that particular region of the DNA. So here, this is called as the footprint of that particular protein on the DNA. That is why DNAs will not be able to cleave the that region where the protein is bound. Okay, so the correct answer is option number two. Okay, so any doubts in this question? So, if no doubts, then we'll move on to the next question. DNA from fossilized samples is extracted. Okay, just a minute. So, what will happen? So, when what we do in case of DNA's footprinting is, this is a double stranded DNA. Okay, so in this case, first we'll separate both the strands, and then we'll allow the protein molecule to bind to one of the strands only that have a particular site of the protein. Okay, then the other strand, that is a complementary strand, so that we allow the DNAs to chop it off alone. Okay, so now what will happen if we this thing run on a gel, then in that case DNAs will form a structure, or the bands that have a ladder kind of structure. Okay, so as uh, I'll show you the image just now. And then if we in the second lane we run the gel where the protein bound DNA is being treated by the DNAs. So in that case, there would be some region where no bands would be observed. After that, the bands would be observed. Okay. So at this particular region, the DNAs will not be able to cleave the DNA because the protein has been bound to this region. Okay. Now, what we'll see, we'll see it using a sequencer. Okay, so now we'll sequence the gene and we'll identify the sequence of this particular protein where it is bound to the DNA. Okay, so this is the major procedure how we use a DNA footprinting. And this is the gel. Sorry for the contrast, but this is how it actually looks on a page gel. Okay, so do you understand? So let's move on to the next question. DNA from fossilized samples is extracted and amplified with random primers. How will you determine the size of the amplified DNA? Okay, so what we do is, for example, in any scenario when we are doing an amplification on a PCR using random primers, so we'll isolate the genome from any sample. Then using specific primers, we'll do a PCR, that is polymerase chain reaction, and then we'll run it on a gel. Okay, so that gel would be between. So if we have a smaller band size, then we use a gel between 1.5 to 3%. Okay, and we if we don't know the size, so where we are using random primers, so we that can be of any size. So that's why we use a 0.8% agarose gel to reduce the size of the fragments that we get. And how will we reduce this fragment size? Using a DNA ladder that basically says you the particular size of the DNA fragment in terms of length. Okay, so for example, there are generally that we use are two kinds of ladder, that is 100 base pair ladder and 1 kb ladder. So 100 base pair ladder starts from the 100 base pair, then it moves on to 200, 300, followed by till 1 kb. Okay, so this forms bands at these particular sizes. The 1 kb ladder starts from 1 kb and moves up to 10 kb. Okay, so this also forms particular bands, and the PCR gel that we are seeing. So, for example, if suppose this is a 300 base pair band at the ladder, and here between this at this particular region you see your PCR band. So that means the product size will be around nearly 300 base pairs. Okay, so that is why we use agarose gel along with the DNA ladder to reduce the size of those particular DNA fragments. Okay, so the correct answer is option number 4. So any doubts in this question? Okay, if not, then we'll move on to the next question. So likewise, this is the correct explanation and how we reduce the molecular weight by using a plot. So what we do is we plot based on the RF values. So as I said, the RF values are the mean length uh, ran by our particular band of DNA in a gel divided by the difference that moves from the whole gel. Okay, so the length moved by the difference. Okay, so this is how we calculate the RF, and based on those RF, we have the molecular weight of the ladder samples. Then we plot a graph, and we reduce the based on the RF, we reduce the molecular weight of that particular DNA molecule. Okay, so this is the extrapolation of the what we are doing and the analysis. Okay, so now moving on to the next question. DNA is resolved in a non-denaturing gel, while mRNA is resolved under denaturing conditions. Because the options are mRNA has uracil in their sequence, mRNA is sensitive to RNase degradation, mRNA has secondary structures, or mRNA is small in size. So yes, the correct answer is mRNA has secondary structures. Okay. So there are different secondary structures that are present in the mRNA because of different properties of the RNA. So there can be loops, or there are basically tertiary structure and secondary structure, like you have seen in the uh, tRNA structure that has a clover leaf-like structure and then the L-shaped structure. So this is a tRNA molecule. This is the secondary structure of this tRNA molecule, and it can fold upon itself. So that is why while resolving a mRNA on a gel, we use denaturing conditions. Okay. So any doubts in this question? Okay. If not, then we'll move on to the next question. So this is basically a typical mRNA gel that we see after isolating the RNA. So, so this is the, for example, here we can see the in case of a pure RNA that we isolate, we see two bands. One is the 28S rRNA and the 18S rRNA. This is case of a eukaryotic cell. Okay. So, and if there is some contamination of the genomic DNA, then we see a separate band above of all the bands, because genomic DNA is has higher molecular weight. So it will form a band that is upward to all the bands here of the ladder itself. Okay, so this is the genomic DNA, and sometimes it will not move out of the ladder also, well also. So this is the genomic DNA contamination of the RNA sample, and then we would also see a mRNA smear. That means the RNA has started degrading. Okay, so whenever we see a smear in a agarose gel, that means that particular DNA or RNA sample is started degrading. Okay, so this is how we study a mRNA gel in real scenario. So now moving on to the next question. The denaturing agent used in agarose gel for resolving mRNA is formaldehyde, TEMET, beta-mercaptoethanol, or SDS. Okay. Yes. So we use formaldehyde for as a denaturing agent. Okay. Some labs also use bleach. Okay, that is sodium hypochlorite, to denature the RNA as well as the RNAs that is present inside the in the gel. Okay. So the correct answer is formaldehyde. Then we'll move on to the next question. RNase free water is prepared by treating water with formamide, beta-mercaptoethanol, diethyl pyrocarbonate, or SDS. So the correct answer is DPC, that is diethyl pyrocarbonate. So what is the function of DPC is that it degrades all the RNAs present on the solution. Okay, so generally whenever we use water for dissolving the RNA, then we use RNase free water by using the DPC treated water. Okay, then if we are isolating RNA in a particular wood or at any location, then what we do, we just wipe the floor or the compartment that we are using DPC water or DPC ethanol that basically degrades all the RNAs present in that particular location. Okay, that is a major usage of DPC here. Okay, that because RNAs are the enzyme that is present everywhere. Okay, even in the air it is there. So if we have to isolate the RNA, then we have to be cautious in that scenario. Okay, so let's move on to the next question. The reagent used to enhance signal of ETBR stained agarose gel with RNA is D stained with acetic acid solution, D stained with 0.5 molar ammonium acetate solution, D stained with RNase free water, or D stained with methanol and acetic acid solution. So yes, the correct option is option number 2, that is D stained with 0.5 molar ammonium acetate solution. So it helps in enhancing the signal of ETBR whenever we are uh, using the UV radiation. So it will give a bright band when we destain it with 0.5 molar ammonium acetate. Okay, so let's move on to the next question. A protein shows a molecular weight of 70 kilodalton in SGS page and 140 kilodalton in native page. What is the oligomeric state of the protein? What can be the correct answer? Okay, any more options? Okay, so in case of SGS page, what generally we do is we apply the protein first with beta mercaptoethanol and then we run it with SGS and page. Okay, so here we apply with SGS and beta mercaptoethanol. So SGS provides the negative charge to the protein that helps to it to unfold, and beta MA breaks all the disulfide bridges. Okay, and in case of native phase, we do not use any of these things, neither beta mercaptoethanol or SGS. So we directly apply this particular protein on a page. Okay, so this will be its oligomeric state, and this would be its monomeric state. Okay, so if we see this, then the and here we get a 140 kilodalton size, and here it is 70 kilodalton size. So this is the half of what we get in SGS phase. So that means the oligomeric state of that particular protein would be dimeric, because in case of native phase that will be the whole protein. So it is giving a 140 kilodalton ka size, and when we apply all the denaturing conditions, and we are getting 70 kilodalton ka size. So one subunit of 70 plus one subunit of 70 will make 140 kilodalton. Okay, so this will be dimeric in state. So the correct option would be option number 1. Okay, I have one. Okay, so what we see is In case of SGS page, when we do the pre-treatment of a protein, then we apply the protein with beta mercaptoethanol and SGS. Okay, so in that case, what happens to the protein is unfold, and then if it has any of the interactions with other proteins, okay, that has been linked by the disulfide bridges is also broken because of the beta end. So here it will be in the monomeric state only. In case of native page, if we see, we apply the protein without any treatment. Okay, so this will give the oligomeric state of the protein. That means all the subunits that have been bound to that particular protein will be seen in the native page. So here we are getting 140 kilodalton in case of a native page, and here we are getting 70 kilodalton in case of a SGS page. So now, if we see that 140 is just the double of 70, so that means 70 plus 70 will give the 140. So one subunit is of 70, the other subunit is also of 70. These both are linked, and this is the dimeric state of the protein. So that is why two subunits will be there of the protein. So am I clear? Okay. So I have a question for all of you here. Can we deduce how the two subunits of a protein are linked? That means either by disulfide bridges or with covalent interaction or hydrogen interaction. Can we deduce here?
Yes, sir. I think beta-marker ethanol that attacks only disulfide bond. So
Okay. So, but the question is because here we are adding both SGS as well as the beta mercaptoethanol. So, if for example two subunits are bound only by hydrogen bonds, okay, not with any of the covalent interactions, then it will also break. And if it will have disulfide bridges, it will also break because beta mercaptoethanol is also there. Okay, so for that scenario, we require additional experiments also to confirm whether the interaction between the two states is disulfide or only hydrogen bonds or any other covalent interactions. So, am I clear?
so to confirm that we need another experiment, right?
Yeah. So, likewise, we what we can design also. For example, here if the protein is bound by the disulfide bridges, so even if we use any of the denaturing agents like beta me, so sorry, reducing agents like beta me, that will break off the disulfide bridges. But if it is attached by the hydrogen bonds, okay, so if we apply only SGS, that will not break the disulfide bridges. So that will help us to clarify whether the disulfide bridges is there or the any other covalent or non-covalent interaction is there between the subunits. Okay, so let's move on to the next question. So vertical streaking of protein band in SGS page occurs because of overloading of protein sample, underloading of protein sample, uneven migration of the protein sample, or uneven heating of the gel. What can be the correct answer? So here the correct answer is option number 1, and this is how a overloading or the vertical streaking of a SGS page looks like. So as you can see here, uh, the other proteins are arranged in a band fashion, but in this case, in the third well or the middle well, the bands are not perfectly visible, but it is a smear kind of stuff. Okay, so this is because of the overloading of the protein in this particular well. Okay, so generally what we do is we add only a specific amount of protein. For example, we can use 6 microgram of protein in a particular well, so that it will be easily separated in an SGS page. If we add more than that, then what will happen? This is the vertical streaking. Okay, so here we'll not be able to see proper separated bands of those particular proteins, and that will be in a smear kind of fashion. It will be observed. Okay, so this is the case of the vertical streaking in a SGS page. Okay, in case of underloading of the protein sample, we'll see very light bands, not these proper bands. We'll see the bands at a very in that are very light. Okay, and the third option, that is uneven migration of the protein sample. In that case, what we'll see, will be seeing particular patches in the protein gel. Okay, so that will also be not the call. And third is the wait a minute. And the fourth one is uneven heating of the gel. Then in that case, what we'll see the smiles on the gel. So the there would be these kind of bands observed on the gel because of the uneven heating of the gel. Okay, or the buffer is not proper. That means the gel is not running properly. So in that case, we'll see smiles of band like this. Okay, I hope I'm clear with the explanation. Okay, so let's move on to the next question. Appearance of distorted protein bands in SDS page can be troubleshooted by decreasing amount of TEMED and APS, increasing voltage of power supply, increasing amount of TEMED and APS, and decreasing the voltage of power supply. What can be the correct answer? Okay, so the correct answer is option number 3, that is increasing amount of TEMED and APS. So what happens if we are seeing distorted protein bands? That means the gel pores that have been formed are not proper. Okay, and the gel is not cross-linked properly. So the cross-linking agents that are TEMED and APS, so these help in cross-linking of the gel. Okay, so whenever we see protein bands that are not proper, that means like if this would be the original protein band and we are seeing like this or like this, that means the gel is not properly cross-linked. So in that case, we'll increase the amount of TEMED and APS for the proper cross-linking of the gel and formation of the pores. Okay, power supply will have no role in formation of the protein bands, uh, distorted protein bands particularly. The Power supply will have role in forming the shape of the protein bands. So whenever the voltage supply or power supply will not be proper or the buffer will be improper, in that case, as I told you before, we will see like this bands or this bands. These will be seen on a gel. Okay. So is the explanation clear? So let's move on to the next question. Unfolding of a protein can cause dash, which makes its mobility slower in the SDS page. So increase in charge, increase in molecular mass, increase in hydrodynamic volume or decrease in hydrodynamic volume. Okay. Any other answers? Okay. In case of SDS page, uh, what is the major factor for separation of the protein? Is it charge or it is mass? Sure, it is charge. Okay. So tell me what is the role of SDS here?
What is the question, sir?
The role of SDS in SDS page.
It's an anionic detergent.
Okay. So if it is an anionic detergent, that means it will provide negative charge on the protein, right? So whenever we run this particular protein that have a negative charge on that on an SDS page, that means it will move towards positive charge. So the charge on the protein will have no role here. Okay. So in case of SDS page, the proteins will be separated on the basis of their molecular weight. Okay, this is the first thing. Charge here will not play any role in separation. It will help in movement of the protein in the gel because the negative charge will move towards the positive charge. That's it. Okay, here molecular weight is the major separation factor. Okay, now the second scenario, this is what has been written in the books and everywhere, but the second scenario is that whenever we run any protein or DNA molecule on a gel. Okay, so it runs on the basis of its size as well as its molecular weight. And here from size, I'll consider shape also. Okay, so whenever we consider shape as well as molecular weight, this will whole will be considered as a hydrodynamic volume. Okay, so considering this fact, if we have a circle of protein or a square of protein, then circle will move faster as compared to a square or a rectangle, right? So here the shape of the protein will also matter, and this will be included in a hydrodynamic volume. Okay, so now when we see a protein, for example, this is a protein, this is fully folded protein. If we apply SGS on this particular protein, it will be unfolded. That means it will be like this, like a chain. Okay, now its hydrodynamic size would increase because here the shape is open now. Here it was circle, for example, or closed, and here it is open. That means it will take large amount of area and large amount of time to pass through a particular gel. Okay, so if we take consider this fact, the molecular weight remains the same in this scenario. It will not increase the molecular mass because the protein is the same only. We have just opened the protein into a open form, that is unfolded state. Okay, so this will increase its hydrodynamic volume rather than its molecular mass. Okay, so here the correct answer would be option number 3. So, am I right, or am I like clear to everybody? Okay. So, for example, as I have CA, that we consider different shapes. Okay. So, for example, we have a circle shape, we have a square, and we have a rectangle. Okay, these are different forms of a protein. For example, okay, and we run on a particular gel. So, if you can see, the molecular weight of all the three proteins are same. Okay. Now, if we run it on a gel, then circle protein, because it is compact. Okay. So it will run more as compared to the proteins that have some other shape that is more in the volume. Okay. And the rectangle protein, because its shape is different and its like stretched part, it will be forming a band that is above the circle wala protein ka band. Okay, so now what happens if these three have same molecular weight but the shape of the protein is different? So in that case we say that hydrodynamic volume that considers both shape as well as the molecular weight of the protein in context. So in this case, the hydrodynamic volume is more in this case in this protein than this protein. Okay. Now, if we see this case in case of a folded protein, so consider a folded protein to be like this because it is really compact in nature. So when we unfold it with the help of any denaturing reagent, so it will unfold it in a this fashion. So it is a chain of amino acids. So it will open as a string. Okay. So now, if its hydrodynamic volume is for a circle and this will be for a chain, so here the hydrodynamic volume would increase. Okay, but the molecular weight of this particular protein will be same because we have not removed any of the amino acids from this particular thing. We have just opened the protein. So here the molecular weight remains the same, but the shape of the protein different. So now when we run both the proteins, whether it's folded or unfolded on a gel, then the folded protein will move to more length as compared to the unfolded protein. So unfolded protein will form a band much up as compared to the folded protein. Okay. Now the same thing we see in DNA also. For example, we have the plasmid DNA. Okay. So here I'll draw. This is the plasmid DNA, and it also has some of the forms. For example, supercoiled, and then we have the nicked form. So in case of supercoiled form, it will be coiled in such a fashion that it will be forming minimum space. It will be occupying minimum space. Okay. Now, if this any of this part is nicked, so in that case, it will be open like this. So it will not be able to coil upon itself. So whenever we run it on a gel, the particular plasmid DNA, the supercoiled will form the lowest band. Then comes the nicked one, and then comes the linear DNA. Okay, so this is separation is based upon the hydrodynamic volume. Okay, and the molecular weight of these three plasmids is the same. So the separation is every time on the basis of the hydrodynamic volume. So now am I clear?
So how it is increasing, like?
Okay, so uh, you got this point, right? Whenever the protein will be closed, then it will occupy minimum space. Okay,
Yes, yes.
so in that case, if we consider a gel, I erase this first. So in case of a gel, for example, this is a pore size. So this is a pore, and if this particular protein is moving through this pore, will it can flow easily. And if the protein is open, because it is in a chain form, so either the chain can be moved like this, or the chain can move like this. Okay, but in a mixture, it can be both ways. So it will not be able to move like in any of the directions, right? Because its shape is different as compared to the globular or a folded protein. So am I clear?
Okay, okay. Thank you, sir.
So this was the last question for today. If you guys have any doubts in the presentation or in any of the part from week 3, then we can discuss that. So if you guys do not have any doubt, yeah, okay. So there is one question. Because as I explained just now, like in this case, uh, whenever we consider a protein or a particular nucleic acid that we are running on a gel, so in that case the hydrodynamic volume would account for the space that is that the particular molecule is occupying. Okay, because as we say that the simple definition of matter that we say is the anything that occupies space, right? So here, by hydrodynamic volume, we mean that the particular protein or a particular molecule is occupying space in that particular jet. Okay, so if the protein is open, so that means it is in a form of a cylinder. Let's consider, okay, or a chain. So if a chain is moving in such a way that it is moving like this for a pore that is of this shape, so in that case it will occupy more space in moving this particular band. So that is why we are considering that it is occupying more space when it is open. So for that space word, we have a scientific term called as hydrodynamic volume. So, okay, okay. If no more doubts are there, then we'll close the session for today. Thank you guys for joining.